Thanks so much, Chris. I'm really happy to be here today and talk about my most favorite microorganisms, the archaea, for sure. So I hope you can hear me well and see my slides. I'm just moving forward, so this seems to work. Great. So we are talking about archaea today, and archaea are a very crazy type of microbe that is too special to join the bacteria club. So you might know that archaea are prokaryotes, of course, like bacteria, and they look like bacteria, as you can see here nicely in this picture, but they are not bacteria. Based on the different background in evolution, and this is a very simplified tree here, the archaea have evolved separated from the bacteria. So we're looking at three domains of life: bacteria, archaea, and eukaryotes. And archaea have an own unique biochemistry. This is very important to consider also with respect to the methods that we are using to really understand, to detect and analyze these archaea. And in many other ways, the archaea are just special. They are not bacteria and they should not be treated as bacteria. So I think many of you might not be aware that we are coming across archaea in many times and in many situations. And particularly, many archaea have really a high impact on global ecosystems. I'm just showing the example of methane production. So whenever we observe methane production, like in wetlands or in the cattle rumen, it's all about archaea because it's methane-forming microorganisms of the domain of archaea that make these methane. And you know, methane is a very important gas. It contributes, of course, to the gas, the atmosphere, and this is, of course, accelerating the climate crisis a lot. So archaea have an impact on our daily life. Many of you might still have the picture of archaea being extremophilic microorganisms thriving in very crazy environments like volcanic environments or hypersaline lakes. This picture has changed a bit in the last 10, 20, 30 years as we recognize archaea being widely abundant in moderate environments, but also, of course, in host systems. And this is, of course, what I will be focusing on the host systems. But before I go into detail, I would like to span the view a bit on human archaeo research and telling you how this actually all started, at least for my research group. As a postdoc and PhD student, I was working a lot with cold-loving archaea, so archaea from cold sulfidic springs, and I was then invited for doing a postdoc at the NASA Jet Propulsion Laboratory in California. So the idea of these researchers back then was to understand whether we could contaminate Mars with Earth methanogenic archaea. There was this very interesting finding back then that methane is being produced on the Mars surface, in particular when the surface warms up. We can observe that there is methane plumes. And it was considered that it could be a biotic source. So potentially methanogenic archaea could thrive right under the surface of Mars and produce the methane. So if you send now missions to Mars to find life, these missions should not have any traces of methanogenic archaea on them, right? You want to detect them and not bring them with you. So what I did during my postdoc is actually I analyzed clean rooms and spacecraft and was trying to find archaeal signatures on all these systems. So I was basically taking samples from clean rooms. So the interesting point here was that we could indeed find archaeal signatures, but we didn't find many methanogenic archaea. We found many other archaea. Back then they were called Crenarchaeota, and now it's the group of Nitrosospharia. This was actually a group of archaea that was associated to soil or aquatic systems, and we could not really understand why we would constantly find them in different clean rooms, spacecraft clean rooms all over the world. You must understand that clean rooms are actually very clean, right? And the only item that cannot be cleaned properly is the humans going in. And so we had this final idea that maybe these signatures actually stem from human skin. Overall, in spacecraft assembly clean rooms, you see a lot of signatures of human-associated microbes because you exhale microbes, you touch surfaces, even if you wear a special garment. So we did a bigger study. We analyzed hospital surfaces, and we also observed and analyzed samples from human individuals. So we took samples from skin. And this was actually the first attempt to do so. And we were able to find same signatures on these humans. And we also could visualize like cockroach-shaped archaea, potentially archaea in these samples. So this was actually the initial point for me to get really interested in the archaeal diversity in the human microbiome and to start all the studies that I'm presenting partially today. I must say that there were a lot of doubts also from the scientific community whether these archaea I just showed from skin would really exist. It was only signatures, it was only fish pictures that we could show at this time point. And I wanted to really dig deeper into that, also solve the methodological issues that we had at this time to really detect archaea in the human microbiome. So I told you that archaea are super different because of their different evolutionary path. And of course, there's a lot of problems to solve if you want to look into this domain of life more specifically. First of all, they have a different cell wall. Their cell wall is much more strong and can be opened with different enzymes and different mechanical treatment. But back then, like 10, 15 years ago, a lot of kits that are used for DNA extraction contain lysozyme, and lysozyme would not open an archaeal cell at all. So we had to tackle this. How do we actually open an archaeal cell to get the DNA out? Also, back then, a lot of amplicon sequencing was done. We had to check the primers, and the primers would not really fit to the Archaea 16S RNA gene. Also, the database is a big issue still today. There is not much data about Archaea, and it's difficult to get genomes and to get information on Archaea. Usually, there's a lot of misclassification that we find and really problematic background here. Also, we don't have many isolates, right? They are not too much present in culture collections. There's not much possibility to work with these archaea. Also, another point is actually that not a single archaeon is known to be a pathogen, not for plants, not for animals, and of course not for the human. So, and of course, this comes also with the aspect that no funding is available also for this research. Other problems were that other archaea researchers didn't have access to human samples. That was, for me, it easier because I'm at a medical university. I can easily access human samples. And also archaea, we have to be careful about this. Sometimes they are really underrepresented and they're really difficult to kind of get them out from a certain situation. So these were all the problems we were actually struggling with, and we wanted to address all of them. Those with an orange dot, I think we have a clear plan how to deal with this now, and a lot of new aspects were found just by optimizing the methods with respect to archaea research. So I mentioned already the problem about the primers and the DNA extraction. And as soon as you use the correct primers that would also see archaea properly, you will see that there is a huge diversity of different archaea all over the human body. You would recognize them, of course, in the gastrointestinal tract and the skin, nose, of course, also in the lung and the upper respiratory tract, of course, as well. So on all these locations, similar to bacteria, archaea come with a different biogeography. So you have different archaea, of course, in the gut than you would have on skin. On skin, I already told you, these are nitrosospharia, ammonia oxidizing archaea that we have on skin. They're obviously taking urea that is produced by sweat, in the sweat, and they use it for energy gain. In the situation in the gastrointestinal tract is somewhat different because you have anoxic conditions, and there we mainly have methanogenic archaea all over the place. So the diversity was surprising that we found, and this actually also brought us to the wish to really produce bigger data on the archaeome. So what we did here, and this is just the pipeline of work that we did for this analysis of 1,000 different archaeal genomes, we mined archaeal information from numerous datasets, metagenomic datasets. We assembled the genomes and we put them together in a meaningful picture. Overall, more than 1,000 different archaeal genomes were obtained. They code for 28,000 protein clusters, and these protein clusters were analyzed with respect to the present viruses or horizontal gene transfer, metabolism, and so on. But we could also identify different strains and species, and now we have this bigger data set that is available. So, of course, also we need to work on the optimization of the cultivation. It's very important to find new pipelines and develop new pipelines to be able to isolate these microorganisms as well. We're focusing on Methanobrevibacter species. This is the most abundant archaeon in the human gut. And we have now established a pipeline where we basically can isolate all the Methanobrevibacter from a single fecal sample. I'm coming back to this a bit later. So, of course, you want to now know which archaea do we have in the gut microbiome and how many of them are actually there. So, what are we talking about? So when we go back to the 1980s, we had very few isolates already available in culture collections. So it was already known that archaea can exist in the gastrointestinal tract, and it's mostly Methanobrevibacter smithii species that had been isolated from human fecal samples. This is the smithii type strain. I hope I'm correct, type strain, which was isolated in the early 80s. And since then, it's basically waiting for more detailed analysis, and is only a handful of researchers all over the world, so only a few groups that really focus on studying the characteristics of this methanogenic archaeon. Now the picture is much broader. As I told you, we have these 1,000 archaeal genomes, and when you look at the diversity of these genomes, you would recognize that indeed Methanobrevibacter species are the most abundant amongst these. So they make 85% of all the genomes that we have. You would also recognize Methanosphera, Methanocopusculum, crazy guys that we don't really know what they're doing. You would recognize also halophilic archaea that are very abundant also in Asian communities and populations. And this is a big group here, Methanomycelicoccales, methyl using archaea that are also very interesting, very widely distributed as well, but we do not really have a grasp on them much. So Methanobrevibacter is quite abundant. Basically, these guys we can really find in almost all healthy microbiomes. They can make up 4 to 4% of the microbiome, so they are really quite abundant amongst the different bacteria. There's also Methanosphera. It is an interesting archaeon as well. Usually it's associated a bit to disease and inflammatory situations. It's not very prevalent, and also the abundance is not really high. So the immune system recognizes this archaeon quite well, and it seems to have to do something with inflammatory processes here. Metanomycelicoccus is one representative of this pinkish group here, and this one specifically is not very abundant, not very prevalent, but the other group in general, it is. So when we look at the Methanobrevibacter, as I just mentioned before, that Methanobrevibacter smithii is the most famous one in this group. But based on the genome analysis, and also other people before that had already recognized that there is another group of Methanobrevibacter species, which we now name Methanobrevibacter intestini. We are in the frame of describing this new species. We have it in culture, so we can really analyze it quite well, and it comes with a lot of different traits. It has a higher genome size, and many other different aspects are different compared to the Methanobrevibacter smithii. The important thing here is that based on the 16S gene, these two species would not be possible to be separated. So we could not recognize that it's smithii or intestini in our samples that we are analyzing. We have to look at the genomic properties and really to identify which one of both is actually present in which situation. So, and this is it. This is Methanobrevibacter intestini. We're working with it right now. We have it in the culture. It's fun to work with. It's somewhat different to Methanobrevibacter smithii, and there's a lot of more details that will be hopefully be coming in the next years on this specific microbe. So, what is actually the function of these archaea in the gastrointestinal tract? Let's look a bit deeper into what they actually do. So, Methanobrevibacter species and methanogenic archaea in general are actually at the very, very end of the food chain. So, as you all know, the food chain starts off with complex organic compounds, lipids, polysaccharides, proteins, and you have different types of fermenters that use these compounds and break them down to little pieces of hydrogen, CO2, acetate, formate, methyl compounds, all that stuff. And this is actually what the methanogenic archaea need for their living. So they are taking all these compounds and producing methane out of it. So what you recognize here is actually that the methanogenic archaea, Methanobrevibacter in particular, is very much dependent on the activity of the bacteria. And the bacteria also depend on the methanogens because they are consuming all the potential inhibiting metabolites from this fermentation process. So there is an interdependency of bacteria and the archaea in the food chain. So these networks can be found in different places all over the body. So, for instance, in the mouth, you will find Methanobrevibacter oralis interacting with microbes, also with microbes that have something to do with periodontitis. And it's actually also known nowadays that these methanogens actually support the bad activity of these microbes, and they really support the driving of periodontitis. And there's, of course, also other locations where we find methanogens, Methanobrevibacter smithii here in the gastrointestinal tract, Methanosphera, and so on, and they produce methane. And this methane, interestingly, also affects us as humans because it's causing constipation. So it slows down the intestinal peristaltic activity, makes it less effective, and so we suffer from constipation if we have a lot of archaea in our gut. Methanobrevibacter species, as I told you, are very abundant in these host microbiomes. Many other archaea are there as well. You will find methanogenic archaea basically all over the place, in the human, diverse animals, of course, and also in plants. So, in particular, Methanobrevibacter species are really prone to interact with host microbiomes. And you see here very strongly Methanobrevibacter. You will find them in a diversity of mammalia, in birds, in insects, and in plants as well. So this is really an important microbe in host microbiomes and interacting strongly with a variety of bacteria. So, and one interesting aspect, I told you before, the archaea that we know of today, they are not pathogenic, not at all. They support sometimes bacteria that can act in a pathogenic way, but they are not actively contributing to human disease in such a way as we know it from the bacteria. So, what do we know about the interaction of human health and disease with archaea? So first of all, let me go back to this Archaea genome catalog that we had. I told you 28,000 proteins clusters that we observed. Interestingly, more than 50% of all the hypothetical proteins that we identified are completely unknown. So we don't know what these proteins are doing. But what we can do is we can use this protein catalog to predict things. So we can make associations with the metadata that have been observed for the different individuals. And what we find is that based on this protein catalog, we can predict something about the lifestyle of the individuals. We can predict where these people come from, continent, country. We can predict the health status, the age group, the sex, and also the BMI. Of course, this is only associations, correlations, and doesn't mean anything about mechanistic backgrounds, but it means that these archaeal proteins are meaningful. They are meaningful for the phenotype of the individual, and this makes it very interesting to go deeper into this. What we know at the moment with respect to different diseases, there's some aspects that are very interesting. Periodontitis, I already told you. So there is an association of methanogenic archaea, Methanobrevibacter being increased in periodontitis samples. We also know, of course, about the constipation. This is very strong information. This has been shown mechanistically. Methane slows down the peristaltic activity. There's also an interesting aspect with respect to SIBO. I'm not sure if you have heard of this disease already. It's small intestinal bacterial overgrowth. So some people have too many bacteria in the small intestine. And this comes with diarrhea, with bloating, and they don't feel very well. So this usually has to be treated somehow. There's a subtype of SIBO which is called IMO, intestinal methanogen overgrowth. And these persons, they produce a lot of methane really fast after intake of a certain food, and they also suffer again from constipation. So this is a very interesting overabundance of methanogenic archaea that needs to be further addressed. Other points, other literature has reported, for instance, an appearance of in multiple sclerosis, so a higher abundance of archaea in multiple sclerosis. We have addressed this in a recent metadata analysis and meta-analysis, and we could identify that actually multiple sclerosis is not coming with a higher abundance of archaea. It's rather to do with the medication that is given to the multiple sclerosis patient. So this is something we could actually deny. This hypothesis was not correct. An interesting aspect, however, is that usually methanogenic archaea, in particular Methanobrevibacter, higher abundance comes with a lower BMI of individuals. This is also something that we know from cows, from cattle already. So if they are really having methanogen growth, and they have because they produce the methane, they're losing about 12% of the energy intake just by the methanogenesis. So they're losing a lot of the energy. And this is obviously similar in the humans as well. So if there's a lot of methanogenic archaea in the gut, these individuals also have a lower BMI. I did some calculations, so it comes down to a cookie per day, but still it's something, right, and could explain this quite difference. If you think that having these methanogens in the gut, actually there would be a lot of bloating and gas production. Yes, there is, but it would be even more if they would not be there, because stoichiometry tells us that there's 4 units of hydrogen and 1 unit of CO2 for 1 unit of methane that is being produced. So actually the gas volume is reduced by the activity of the methanogenic archaea. I would take some little few minutes to talk about the phenomenon of the human cows quickly. So about 40% of the North American and European population exhales quite substantial amounts of methane. So these are human individuals that are acting a little bit as a cow. We jokingly call them human cows. This percentage is lower in Asian populations and higher in African populations, where we see about 80% of individuals producing measurable methane in their breath. And this is interesting because methane is a very specific component to methanogenic archaea. So there is no other bacteria that can actually produce, or microbes that can actually produce methane. So we wanted to understand what is actually the difference between the human cows and the non-cows. The cows you will see in blue color here. So the human cows, they come with 1,000-fold increase of Methanobrevibacter in their gut. The archaea make about 2% of the microbiomes. This is a really substantial amount. They produce methane that we can measure, more than 5 parts per million in their breath. They come with this lower BMI I just mentioned, and usually these people show longevity and they're having a healthier life. So this is really an interesting cohort. The low methanogen phenotype is the opposite. So we have 1,000-fold decreased abundance of Methanobrevibacter. The breath methane is not detectable or super low. We wanted to go deeper a bit more in the differences of these different cohorts and did some microbiome analysis. What you see here is that the microbiomes are completely different in these two. So first of all, in the human cows, we see that the cycle of or the food chain is directed towards methane production. So everything is prepared to produce CO2, hydrogen, acetate, formate, so everything what the Methanobrevibacter is using. And it's taking this and it's producing methane. Methane usually leaves completely the body and the microbiomes. It's not of any use. It only causes the constipation thing, but apart from this, it goes away. So what happens in the microbiome is that you have a constant pull factor towards the methane production, and you're losing basically a bit of energy, and everything is going towards methanogenesis. In the other microbiomes, this cycle is kind of active all the time. So the CO2 hydrogen is being produced, but it can also be again put into acetate and so on. So these energy and carbon compounds, they stay more in the cycle and can be consumed by more bacteria here. So the situation is somewhat different. We see also different phenotype in the human cows. They usually have a higher uptake of dietary fiber. They produce more short-chain fatty acids like formate, acetate, propionate. We can also see differences in their urine. So they produce more succinate citric acid, and they tend to be vegetarians. So they are indeed rather cows. In the non-cows, we see that the dietary uptake of B12 is higher, and they also eat more fat. So again, let's look at this. We see the different metabolites in stool in the human cows, formate, acetate, propionate. We see similar things in the urine. And of course, we tested also our cultures that we to see whether these products are also there. And again, in the cultures, we also found formate, acetate, succinate, and again, propionate. So it's indeed the archaea that are able to produce these compounds. At the moment, we don't know what this means. We don't know, is there direct signaling, for instance, occurring, or does this mean anything to the immune system? This is something we have to study in the next weeks and months and years. So I'm quickly coming back to the archaea on skin, right? You remember how it all started by detecting archaeal signatures on skin, and we did not give up on this topic as well. And we did a bigger cohort study in the last years, looking at 47 different individuals. The longitudinal way was done, and we again found the same ammonia oxidizing archaea signatures, particularly in the forehead of all of these individuals. This was independent from the season. It makes about 0.2%, so not as abundant in the gut as we observe it. And Nitrosocosmicus is the one that we observe here mostly. This genus is the one that appears most often across these individuals. And the interesting thing is that Nitrosocosmicus is also embedded in the microbiome network. So it's obviously co-occurring with different bacteria, which actually tells us that there is maybe also metabolic exchange and more strong embedding in the overall microbiome. Well, the final proof to really make sure that these archaea are really there is, I think, the cultivation from those samples. And our collaboration partners in Vienna, Christa Schleper and her group, have done a fantastic job in getting 4 stable enrichment cultures obtained from different individuals of these ammonia oxidizing archaea. They're growing, they're doing their job, ammonia oxidizing, and also their genomes that we are analyzing at the moment show adaptations to the skin environment. So they're really there, they're active, we can isolate them, we can now study them in a bigger context as well. So just remember the picture that I showed you before from 2013, where we identified cockroach-shaped archaea here. Obviously, these were already the Nitrosocosmicus species that we have now in culture. So, and we put this all together here in this final tree. So we have the isolates here that you see here, and this is the the sequences that we obtain from our sequencing approach from skin directly. And you see this is all the same cluster. This is definitely something that is growing on our skin and is constantly present and detectable. So to come to an end, of course, we need to think about future perspective, and we want to really enter mechanistic studies now. And for that, it's very, very important to have these cultures at hand. So we really need more cultures. We need more species, strains for mechanistic studies. And here I'm just showing how we did this in the past very successfully. So we use samples that we pre-screened from donors for methane production in their breast. So we identify who is a human cow and who is not. And these we use to really go into this cultivation scheme. We have optimized the medium. We have done in silico metabolic modeling for that. We use also partner bacteria, so we have an idea which bacteria they really like to interact with. And this we also add to these cultures. And we also use a certain fluorescence these archaea have to do cell sorting through FACS and so on and so on. So we really, it's a laborious way, but basically we are now able to isolate archaea from each individual. For here, we tested for 16 different individuals, and from 14, we were able to obtain their Methanobrevibacter species, except Crohn's disease patients. These are different and special, and from them, we were not able to isolate any of these methanogenic archaea. So by this approach, we identified 8 novel clades of Methanobrevibacter, and they come a lot with different genome peculiarities, which have to be analyzed now. And of course, we want to do mechanistic studies with all of them. So this was the work I presented from many people in my lab team. So Alex, Steffi, Victoria, Christina, Roxy, and Victoria have done an amazing job. You can read about their work. I presented only glimpses of it and parts I did not mention at all about the aging interaction or the vesicles that we identified from these methanogenic archaea that might act on the human surfaces. So please go ahead and read their papers. They will be very happy about this. So one final aspect, and this is really intriguing, archaeal pathogens. We have done a lot of research now and others as well. We have not identified a single archaeal pathogen to date. I'm showing here Carl Woese, who has actually described the domain of archaea tens of years back. He never received a Nobel Prize for that, which is quite surprising, but there is no Nobel Prize for biology. It's only a medical Nobel Prize. And well, maybe he was not given the Nobel Prize for medicine because archaea were thought to have no medical relevance. This has changed a bit, but still we have not identified a pathogen. And there's a lot of discussion why this is the case. Maybe they're dependent on bacteria, they're growing too slow, many aspects. And this always makes fun to think about why archaea cannot be pathogenic, although they are so abundant and prevalent in host microbiomes. With that, I actually want to close. I have to thank many people and many funding givers, of course, as well. One note, we are hiring. I'm looking for a postdoc. So if you're interested, please contact me. Also, PhD position will be open in the next few weeks. Let me know. So thanks again for listening. I'm looking forward to your questions. Thanks a lot.